Hello everyone. The next topic we're looking at is enzymes, and you probably remember bits and pieces from GCSE. So when you think of enzymes, you maybe think of proteins that speed up chemical reactions. Now I'm just going to jot a few things down here. You don't need to copy them down, but every now and then I'll talk over things that come up in your notes, and you might want to pause the video or stop it and just get those bits of your notes filled in. Or you might want to watch it all and then just go back to your textbook and try and fill in your notes that way, whichever suits you better. But we're just going to spend the first few minutes recapping what we know about enzymes. So speed up chemical reactions. And you probably remember things like pH and temperature can affect how they work. And you know that enzymes are found in your digestive system. And really they control all of the reactions in your body. Now, there's a word which is metabolism. And I'm sure you've probably heard about it, but metabolism really is the name for all of the chemical reactions that occur inside the cells of your body. In fact, the cells of all living organisms. Now, sometimes the reactions will take small molecules and build them up together to make larger molecules. So, for example, if you have two alpha glucose molecules and you join them together by a condensation reaction. Hopefully this is jogging your memory. You should remember that you get one molecule of maltose. Okay, so we've got two disaccharides. Sorry, that's wrong. We've got one monosaccharide and another monosaccharide, and we're ending up here with the disaccharide. So we looked at that in the last topic, and we know that we can go down the way and build up a larger molecule. Maltose is the larger molecule. Or we could go the other way and break up maltose into its constituent monosaccharides, and hopefully you'll remember that reaction is hydrolysis. We need to add a molecule of water in there, and we get a water molecule coming out of the condensation reaction. So that's a little bit of revision there for you. Now, if we're going from the glucose and glucose, and we're joining two relatively small molecules into one larger molecule, so going in that direction, that type of reaction is described as being anabolic. And the process is anabolism. Okay? If you're going the other direction, from the larger molecule into the two smaller ones, that reaction is catabolic. So it's a catabolic reaction, and the process is catabolism. Okay, so anabolic is building up. You've got two smaller molecules making one larger maltose molecule, so that's an anabolic reaction. The way I remember that is sometimes bodybuilders take anabolic steroids to build up their muscles. Now, they shouldn't do that, but some of them do. But anabolic, building up your muscles, you're going from two small molecules or three small molecules or any number of small molecules and joining them together to build up a bigger molecule. And catabolism is the opposite way around. So if we look at these two diagrams, what's going on here? Well, here, these are the substrates. Remember, in a chemical reaction, what you start off with is your substrate and what you end up with is your product. And hopefully from year 11, you'll see the enzyme molecule is the same at the beginning here as it is at the end here. The enzyme molecule is unchanged. So we've got two separate sets of diagrams here. So if we look at the top one, we've got two substrate molecules. Okay. And at the end, they've joined together to make one larger molecule. So we've gone from some smaller molecules into one bigger one. So that reaction is an anabolic reaction. And as your notes say, that's a build-up reaction where larger molecules are built up from the smaller molecules. So the example in your notes is the synthesis of proteins from amino acids, but you could also have synthesis of polysaccharides from monosaccharides, or as we looked at earlier, the synthesis of a disaccharide from two monosaccharides. The reaction at the bottom here, our substrate is a bigger molecule and it's been broken down into smaller products. So that's the opposite. So that's a catabolic reaction. Now, there are thousands and thousands of metabolic reactions that take place in your body every second. So there's reactions to be making new cells, to make enzymes that are needed for respiration, to really to control the speed at which those steps in respiration occur. You're making all sorts of hormones, all sorts of growth factors and so on. And each one of those reactions is controlled by a separate enzyme. So, as we said, they're enzymes. I suppose a definition would be to these diagrams here, we've said that the enzyme molecule doesn't change shape. And that means the enzyme is there at the beginning and then at the end in exactly the same form. And so the enzyme can be reused over and over again. And we've said that the molecules that are reacting are called the substrate molecules. So I've labelled them up at the top here. And what you end up with at the end is called the product. So down here, these are your substrates and these are your products. So I suppose what you could is and an arrow. And really all I'm expecting you to write substrate goes to product. Now, we said that enzymes are made of protein. And when we think of the structure of protein, we think of primary, secondary, tertiary, and some proteins have got more than one polypeptide chain, and so they've got a quaternary structure. So what do we mean by the primary structure? Well, it's the order or the sequence of the amino acids in the chain. Then we said that there will be some hydrogen bonds which will form and cause this chain to coil into an alpha helix or to fold into a beta pleated sheet. And if, for example, you've got a sulfur molecule there and a sulfur molecule there, those two molecules will attract each other, and so you get a disulfide bridge or bond forming, and that will cause the alpha helix to bend. So the primary structure is the order of the amino acids. The secondary structure is the coiling into an alpha helix or a beta folding into a beta pleated sheet. The tertiary structure then is the bending and the folding of the secondary structure into a specific three-dimensional shape. Enzyme is a biological catalyst. That speeds up the rate of a metabolic reaction. Okay, so that's what an enzyme is. It's a biological catalyst that speeds up the rate of a metabolic reaction. And if we go back on here, now if we look at this diagram here, this is, we look at this diagram, what we've got here is our polypeptide chain, and you can see how it's folded and twisted, and we've got a three-dimensional or a globular protein. And the shape of it is determined by its tertiary structure. Now, hopefully you remember from GCSE that there is one part of the enzyme that is the most important, and that is the active site. And that is where the substrate fits into. And for the substrate to fit in there, the substrate has to be complementary. Hold on, I can't spell here. That means that, well, if we look, if that's the shape of the enzyme, the substrate has to be that shape there in order to fit in. And we said that it's complementary in shape to the active site. So this is the active site here. It's part of the enzyme. And the substrate fits into it. Now, an enzyme, if I draw an enzyme like that, if you've got a series of substrate molecules here, so we've got 4 substrate molecules here. This is the active site. Number 1 clearly isn't going to fit in because it's not complementary in shape to the active site. Number 2 isn't going to fit in either. Neither's number 3, but number 4 is the right shape. It'll fit in to the active site. And we say that enzymes are specific. And we talk about this term enzyme specificity. And that's the term which describes the fact that each enzyme is specific to a particular substrate. So these are our substrates. And only number 4 fitted in. And so we say the enzyme is specific. There's only one substrate that is an exact complementary shape to the active site. If you've got 1,000 reactions occurring in a cell, then that means 1,000 different reactions occurring in a cell. That cell needs to have 1,000 different enzymes. So enzyme specificity, it explains that an enzyme is specific to a particular substrate. There's only one substrate that's an exact complementary shape to the active site of the enzyme. Okay? Now, enzymes and substrates actually work. So if we draw an enzyme here, and these are our substrates, Okay. The first thing that has to happen is that the enzyme and the substrate, now I'm using shorthand, you can't, you need to write out the proper words. Enzyme and substrate must collide. So these reactions are happening inside the cytoplasm of your cells. It's a sort of fluid, gel-like substance, so there is liquid there. Molecules are moving in liquids, and so the molecules will be moving just in random directions, and so really it's a bit of luck. But the enzyme and substrate have to collide in such a way so that the enzyme and substrate fit together. So in other words, it's the substrate that fits into the active site of the enzyme. So what do you have there? This is now called an enzyme substrate complex. Okay, so that's important. That is called an enzyme substrate complex. Now, so obviously then the enzyme and the substrate are going to interact. If it's an anabolic reaction, which is what we had there, so that's an anabolic. The substrate molecules are orientated in such a way to facilitate bonding. So in other words, the enzyme holds them in such a way to make bonding more likely. So we're saying so enzyme orientates the substrate in such a way to facilitate bonding or to encourage bonding. If it's a catabolic reaction, so in which case this is one large molecule, the enzyme really is going to kind of put pressure. It's going to push down on this and put pressure in such a way so as to sort of distort it and put strain on the bonds to facilitate the bonds breaking. So the enzyme puts strain Okay, so anabolic reaction, up here we've got two molecules. The enzyme orientates the molecules so that it's going to facilitate bonding. If it's a catabolic reaction, the enzyme is going to put strain on the bonds of a substrate to facilitate the breakdown. So in other words, to make it more likely that it's going to break down. So let's get back to our anabolic one. So after a while, the bonds will have formed, and we've now got one larger molecule, which is now our product. And this is still our enzyme. So this now becomes our enzyme product complex. The product no longer is an exact fit to the active site, so the product is released. And if the product's released, the enzyme looks the same as it did at the very start. The enzyme is unchanged and it's ready to catalyse the same reaction over and over again. So basically the enzyme is reused or recycled. Don't know what happened. Now, if we look at this diagram here, we're going to put some labels on it. So we've got one large molecule at the beginning. So this is our substrate because we're starting with it. And these are therefore our products. And down here, this is our enzyme. At the end, you can see the enzyme just looks the same as it did at the beginning. So substrate and the enzyme collide, and then the substrate fits into the active site. So suppose we could label this as the active site. So the second diagram, we've got the enzyme substrate complex. Now, I'm running out of a wee bit of space here. See if I can make it smaller. No, maybe not. Okay, so that's the enzyme substrate complex. And then I'm sure you've worked out then this is the enzyme product complex. I hope your diagrams are neater than mine. There we go. So the enzyme looks the same at the beginning as it does at the end. We start off with our substrate. Substrate fits in to the active site of the enzyme. It's complementary in shape, and you get an enzyme substrate complex being formed. This is an anabolic. No, it's not. It's a catabolic reaction. We'll just write that at the top. Because we're starting off with one large molecule and breaking it down into smaller ones. Catabolic reaction. So, in the diagram number 2 here, where we've got the enzyme substrate complex, the enzyme is putting strain on the substrate molecule to facilitate the breaking of the bonds. And you can see in the third diagram that we've now got 2 smaller molecules, and therefore that's our product. And it gets released in step 4 or diagram 4, gets released as our products, and we're left with our enzyme ready to go again. The whole point of this, well, any reaction has a energy barrier that must be overcome. It's a bit like if you have, now your notes of this diagram here, if you've got a stone or some kind of a rock or a boulder and you want to roll it down that hill, so that's your, we'll say that's your substrate. And when it rolls down the hill, it breaks into different bits of rock there, which would be your product. To move that rock down there, you need to push it up over that little bump, and then it will fall down. Now, in a catabolic reaction, if that's the potential energy of your substrate molecules and this is the potential energy of your product, we need to add energy to activate the reaction, and then an energy will be given out during the reaction. This energy here is called the activation energy. Okay, so that energy there is the activation energy. What an enzyme does, it actually reduces the activation energy. So instead of having to give that amount of activation energy, the graph would look more like that. And so that would be the activation energy. With the enzyme in red, the activation energy in black is without the enzyme. The graph notes shows a lot more neatly than I did, but that's what we're saying. Without the enzyme, they've got it in red. Without the enzyme, this is the amount of activation, that's energy that's needed. With the enzyme, it's much smaller. So the activation energy is like an energy barrier that has to be overcome before the reaction can take place. So we said that amount of energy is called the activation energy. When we go to do reactions in the lab, we normally get a Bunsen burner out and heat the chemicals to get them to react. Obviously, your body, that can't happen. That would be unsuitable. High temperatures would be unsuitable in your body because so much of your body is made of proteins. And hopefully you remember from GCSE that high temperatures will denature protein molecules. So that's why requiring high temperatures for reactions in the body wouldn't work. Now, the bit at the bottom of the page, it says enzymes work by lowering, and this is what you need to add in, lowering the activation energy. And that means that your body doesn't need to increase the temperature or heat them up. We don't need extra heat to allow these to happen. So therefore, reactions do take place at 37 degrees Celsius inside your body. Okay, now the graph in your notes maybe isn't terribly clear, so you probably want to add this dotted line in here and these dotted lines in here. It might not have come out in your notes. Now, there are two models of enzyme action. Now, you probably remember the lock and key hypothesis from GCSE. And that was put forward to explain why enzymes were specific. Basically, it's saying that the active site of an enzyme is a particular shape, and only one type of substrate molecule will be complementary in size and shape to fit in exactly. So in your notes that it says, the theory was put forward to explain why enzymes are specific. It proposes that the active site of an enzyme has a complementary shape into which the substrate molecule fits exactly. And the exactly is important. So you've got this diagram here. Well, you've got a slightly different shape of substrate. We're going to add some labels. So this is a substrate, the enzyme, and that's the active site. So the enzyme is exactly the same. And in my diagram, I've got this wee bit here sticking out. I suppose if I colour it in, it makes it a bit neater. That's our enzyme substrate complex. There's our enzyme. and these are our products. So the substrate and the enzyme are exactly complementary. So what you should write at the bottom or at the side there is the enzyme fits around the substrate exactly. So it's an exact match for the substrate. If we look at this, the lock and key hypothesis, the substrate shape and the active site are an exact fit. Substrate fits in exactly into the active site. The induced fit hypothesis was developed later on. It's really a more updated version. And if we look at the diagrams then, so if this is our substrate, that's our active site. And obviously this is the enzyme. You'll see that the substrate is not an exact sort of complementary fit for the enzyme. So what happens is when the substrate starts to bind to the active site changes shape and actually molds itself around the substrate. So we've got substrate in there, and this will actually mold itself around the substrate. So this is our enzyme substrate complex. Now, of course, I mean, we could have drawn in another diagram where we've got our enzyme product complex. We're going to skip that, just like we did the last time. We're going to skip it. The products are released, but actually what happens there is then the active site goes back to how it started. Right, I'm messing this up a bit, but you get the idea. So there's our enzyme should look exactly like it did in the first one, because it can start all over again. The thing to write here is that the enzyme's active site molds around the substrate, and that's at this stage here. So somewhere down at the bottom, you've got a bit of space underneath it. If you just say the enzyme's active site molds around the substrate, forming a precise fit. So that's the key difference. If we look at this, the substrate isn't an exact fit for the active site, but whenever the substrate starts to bind in here, then the active site molds around the substrate, forming a precise fit. And as it molds around, again, in this case, it puts strain on the bonds, and that will break the bonds and therefore lowering the activation energy, and the reaction takes place much more easily. As the products are released, the active site returns to its original shape. As we said here, it goes back to this shape here. Now, the last thing we're going to look at now is cofactors. So, basically, a cofactor is a non-protein part attached to it, and that non-protein bit is called the cofactors. So we've learnt that enzymes are proteins, but now we're saying most of them have this extra bit attached onto it that are non-protein. Now, a little bit of revision. When we looked at proteins, we learnt about conjugated proteins. And we said that proteins that had a non-protein part attached, like hemoglobin has got a heme group that contains iron, it's an example of a conjugated protein. So we're saying most enzymes are actually conjugated protein. They've got a non-protein prosthetic group, and it's called a cofactor. And a cofactor is something that the enzyme requires this to function properly. And it's usually because they have some kind of effect on the shape of the active site, or else they actually are part of the reaction. Okay, so there are two cofactors. First ones are called prosthetic groups. So this is a special type of cofactor. So heme, for example, is present as a prosthetic group in catalase. So again, it's a non-protein group. As we know, that's what prosthetic means. But in this case, it's a non-protein. It's inorganic. That means it doesn't contain carbon. And they are permanently attached to the enzyme. So we said that iron, or heme group, is attached on, well, part of the enzyme catalase, and it's always there. If it's not attached to it, it's the enzyme will not work. It's permanently attached to it. Okay. The other type of are non-protein and inorganic. Prosthetic groups are always attached to the enzyme. Factor is coenzymes. Okay, so again, they're non-protein groups. They're organic, so they contain carbon, and they are not permanently attached to the enzyme. Okay, their function is to help the enzyme and the substrate bind with each other. So sometimes, well, if we look at this next diagram, coenzymes, well, yes, they are non-protein, but they're organic, whereas coenzymes are not always attached to the enzyme. A lot of coenzymes contain a vitamin. Now, you know that if you're lacking in a certain vitamin, you might have some symptoms, and we call those like deficiency diseases. And there's a question there. It says, why do people develop deficiency diseases if vitamins are lacking in their diet? So we're saying that basically metabolism, all of the body reactions, the processes that happen in your body, they're all controlled by enzymes. Most enzymes require a coenzyme to function. So if they don't have a coenzyme, the enzyme isn't going to work. If your diet is deficient in a particular vitamin, then your body can't make that coenzyme, and therefore that enzyme isn't going to work. So that reaction that it normally catalyzes is not going to proceed. And so if a reaction isn't going to work, that means there's something not going to work properly in your body, and you're going to be short of a chemical, and it's going to manifest itself in some kind of deficiency disease. Okay, so we're going to leave it there. We've covered quite a lot of information. Really, you should have filled in, let me see, 1, 2, 3, 4, the first 6 pages of your handout there in Enzymes. You might want to go back over things, or you might just want to get the information from your textbook. Okay? All the best. Bye. There's our enzyme molecule. These are our substrates. Now, if we look at the substrate, they are not the right shape to fit into, if this is the active site. Those substrates are not going to fit into that active site. It's the wrong shape. But if this cofactor fits in, it will modify the shape of the active site. So the active site now becomes this shape, and that is complementary in shape to the substrate molecules, so they can fit in. And we've now got an enzyme substrate complex, and the reaction is going to take place. So if that cofactor wasn't there, you're not going to get any reaction. The reaction will only take place if the cofactor is present. So at the top of the page, we'd said enzymes require cofactors in order to function. So usually they will influence the shape of the active site and really make the active site the right shape to allow the substrate molecules to fit in. Prosthetic groups.